So, yesterday we were, we did a mutagenesis and screen and found some genes that are involved in vulval development, right. And one of them was dominant, and therefore, we discussed that you can't immediately conclude what the gene is normally doing for that particular tissue development. Okay. To give you another analogy, like for example, if someone walks into this room, they will find that I am teaching developmental biology. So, the information that they can safely conclude is that I am probably somebody who knows to teach developmental biology and is teaching developmental biology, can teach developmental biology. But they will not be able to conclude whether there is a full semester course in developmental biology in biotechnology department, and that course in this semester January-May 2020 is being taught by this particular instructor, right. That is what you want to know if you want to understand how a particular biological system functions. Who does that normally is what you need to know. How will you find that information? From a gain of function, like right now someone comes and sees and I am teaching, that would be a gain of function, you are seeing a function. But you do not know that function is the normal function of that department for that semester, and it is regularly run. For that, what do you need to do? So, you need to take that given instructor out of the picture and then see whether developmental biology class is happening or not. And if it does not happen, then you know this is the instructor who teaches that as part of that program. So, that is the benefit of a loss of function. So that's why dominant mutations do not immediately tell what is the role of a given gene for that particular biological process. So, most of the time what a given gene is capable of doing is not that interesting. So, what is really interesting is in the normal wild type function biological processes of a given organism, what are the molecules involved. So, that is what is really informative about understanding how that particular organism develops or lives its life. Okay. So, now we move on. So, we isolated some of the mutants, we called them. So, we found genes, we arbitrarily called it as Lin26, not arbitrarily, like it is lineage defective in the order in which it was found, it is 26th gene. But this does not tell about is it an enzyme or is it a transcription factor or is it RNA binding protein, what does it do and how does it finally make a vulva, how does it contribute to making a vulva, right. So, that we do not know. So, we want to find out where in the 6 chromosomes this is located and what kind of protein it encodes, that is what we want to know. So, that process is called genetic mapping. Okay. So, mapping a given mutant. So, now let us focus on how that is done. There are multiple routes. So, I am trying to go through the most common route that is done in standard genetics. So, the first thing is we should have this first. So, first we need to find out on which chromosome that particular given mutant allele is located. Okay. First you want to identify the chromosome. So, for that what we do is we take a known mutant allele of a gene, known meaning in which chromosome it is located. So, in our particular example, since we have 6 chromosomes, we take 6 different markers, each one well known for its position on, let us say, chromosome 1, 2, 3, 4, 5 and X. Now, you do that Morgan's experiment. Okay. So, try to see the recombination frequency between your mutant allele and that known mutant allele. And that recombination frequency, if you find it they are independently assorting, then you know it is not on the same chromosome. If they show linkage, then you know okay, it is on chromosome 1. Okay. Let us say Lin26 shows a tight linkage to a gene located on chromosome 1 and independently assorts with the rest of the 5, then you know Lin26 is probably on chromosome 1. So, this is called two-factor analysis. Okay. So, recombination frequency between the new mutant and a known marker mutation. Is this clear? It is the same logic as what we have already discussed when we learnt about Morgan's experiment with the black and vestigial. So, what I kind of glossed over there and now we are going to look in detail is the three-factor analysis. Now, we know that the chromosomes are really having millions of base pairs. C. elegans has about 100 million base pairs divided into 6 chromosomes. So, obviously, each one of them is having more than 10 million base pairs, right. An average gene is under 2 kb. So, you need to know out of the 10 million bases which 2 kb is your gene. So, there what you try to do is you narrow down within the chromosome to a particular genetic interval, meaning 2 known positions you select and then the distance between the 2 you call as a genetic interval. So, for that what you do is a three-factor analysis. This is really a very useful analysis. It gives you a very specific position for your gene at the end of the three-factor analysis. Here you are doing the same as the two-factor analysis, except that you are determining the recombination frequency between your new mutant allele and 2 different known markers located on the same chromosome at known intervals. Okay. So, now let us see the result of that. Now, here I am using Morgan's example itself, like we are taking black and we determined the recombination frequency of black and vestigial is 17%. Now, this 17% can be either on the right side of black, okay, so, and each percentage is considered as 1 centimorgan, okay, in memory of Morgan. So, 17 centimorgans to the right of black or it could be to the left of black, right. Recombination frequency does not tell you on which side of a given reference point. So, it is 17%. Now, I take another one known one, let us say this En, then I determine the recombination frequency with the black, then I find it is 9%. Now, this 9% could be either on the left or right. Now, you do a third one, the Cn and vestigial, and you find it is 9.5%. That tells you Cn is between the 2 and vestigial is on the right of black. So, imagine vestigial was in this side, okay, and it is 17 centimorgan on this side. Now, when Cn gives you 9% with black, that is not contradicting your first conclusion that vestigial is on the right side. But when you determine Cn and vestigial is 9.5, then that contradicts your assumption that vestigial is on the left side, and that is how you put Cn as 9 map units to the right of black and 9.5 map units left of vestigial. So, you give a specific position for your new mutation. Here we are assuming black and vestigial as known markers and the Cn is the unknown. So, this is called three-factor analysis. So, three-factor analysis gives a very specific position, but the resolution and accuracy of your conclusion depends on the number of progeny you looked at. How many total progeny and how many recombinant progeny you got. So, it is dependent on that. Higher the number, higher the accuracy. So, once that is done, then we need to do a giant leap. That is, earlier they had to continue this, like what you do is first you take markers that are far apart and therefore your allele is somewhere in between. Then you select newer known markers that are closer and closer and closer, then you narrow down to a really a small region. So, that is very tedious, and that is why gene mapping 10 years or not 10 years, probably 20 years ago took few years to map a gene, but nowadays we do it in a month or so if you are fully on it. So, what people do after this is, okay, this is what we do next. So, before we go into this, I need to introduce what is called a genetic map and a physical map. So, this is a genetic map. Okay, so we haven't taken a rod like structure and used a ruler whose scale is length, millimeter, centimeter, etc., and measured it and marked this 9 centimeter, 9.5 centimeter, etc. We didn't do that. Okay. So, this is purely an imaginary map based on recombination frequency. Okay. There is no physical basis for this. So, this is a genetic map. An imaginary map based on recombination frequency among the different alleles, whereas the actual DNA sequence is called the physical map, and the actual DNA sequence is now, but historically it was not really the DNA sequence. So, you need to understand when I was a PhD student like 88 to 94, even as late as 1990, thinking of sequencing a genome of a whole organism was considered as a foolhardy thought and something not practical, and even if technology permits it, people consider that an enormous waste of resources and one should be doing it. There was a school of thought that existed like that. So, but some people were convinced it is worth the effort and we should sequence. And to get geared up to do the human genome, all the methodologies were developed to determine the C elegans sequencing. Okay. And one of the methodologies that they developed was breaking down the chromosome into smaller bits and cloning them in vector, the same concept as plasmid, except that in plasmid you are having few hundred base pairs to few tens of bases, not even few 10,000s of bases, few thousands of bases. Okay, so the biggest insert you might clone is probably about 10 kb to 12 kb, that size. But there are vectors called bacterial artificial chromosomes, yeast artificial chromosomes, and then even earlier than this cosmids. So, these are big vectors, we do not need to get into the details of their molecular biology, but they take large size inserts. Like for example, you can have 100 kb in that range in yeast artificial chromosomes or in bags. In cosmids usually about 40 kb, 50 kb, that size. So, the chromosome is broken down, cloned in these vectors and transformed into the respective organism. Like for example, if you take bac and cosmid, bacteria, and then you have colonies. Now, in each colony you isolate, let us say cosmid, we will stick to cosmid. So, you isolate the cosmid and using, so cosmid sequence is known. So, the flanking sequences can be used as that sequence information can be used to make primers and use those primers as template for sequencing. So, you do not need to know the insert sequencing from the vector portion, from the vector sequence using those vector specific primers, you get some sequence information into the insert. Now, from that, like for example, you can take one end of, you can do two ways, you could either go by sequencing or simply take smaller fragments from that insert, radio label it and screen the library again to find newer cosmid clones that light up with that probe. Like for example, here if you take the first orange one and that light blue one, now randomly let us say I picked this and in that insert I took the two ends. Let us say I took this end, I made radio labeled probe and screened again that cosmid library, and then I find a new one and then I go and take the other end of that one. Okay, so now you see the logic of how this would have picked this, because they are overlapping. Now I take this one and then find this and then go like this. This is called chromosome walking. Okay. So you walk along the chromosome in terms of identifying the cosmid clones that are sequentially arranged along the length of the chromosome. And then now you make a map. Cosmid 1 is followed by cosmid 2, followed by cosmid 3 and so on, the order of the cosmids, and that is what is a physical map. So what is shown here is a physical map. Essentially, if you determine the sequence, it is actually the DNA sequence. So is this clear? So now in our mapping approach, what we do is we jump from this, knowing that it is 9 map units to the right of black. Okay. From there, now you need to jump to this, where in this cosmid that map unit 9 is. So for that again you need to know the known markers physical positions, and once you know that, you jump from the genetic map to physical map. Okay, correlation of the genetic and physical maps near the gene of interest. For that essentially you need to know this one in physical map, this one in physical map. If you know those two cosmids, let us say black is in this cosmid and vestigial is in this cosmid, then you know your gene is within these cosmids. Now what you do is something called DNA mediated transformation. So now you are going to take each one of these cosmids and introduce into the organism. In C. elegans you inject, and then see which one rescues the mutant phenotype, because these are all wild type. Okay, these are made from the wild type organism. Now you introduce and then see which one rescues. Alright. So here we are sticking with what we discovered. Remember let60, the dominant one that we got. So the genetic mapping, the 2 factor, 3 factor, etc., put let60 between dumpy20. So dumpy20 is a known marker. Okay, and its physical map is known, cosmid this number. And the left breakpoint of, okay, so this is a deficiency. Deficiency means larger deletions of chromosomes instead of point mutations or a few hundred base pair deletions. These are larger deletions. So they are often used to determine whether an allele that you got is a null allele or not. So that we will deal with it if we ever need to get into it. For now, all you need to know is treat this as another known gene. Okay, like dumpy1020 is a known gene. Its genetic position is known, physical map, this cosmid is known. Similarly, this is another known mutant and its genetic map is known, cosmid is also known. Now you take all the cosmids between the two, and these are those cosmids. Okay, this is a hypothetical. So we are not listing all the cosmids that may be in between the two. Then you do injections. Now you inject this one and then you find it rescues dumpy20 but not let60. Okay. So somewhere in it dumpy20 is covered but not at let60. Then you go to this one, then you find it rescues both. It is kind of telling you that maybe dumpy is at this end and let60 is further to the left of it, and that gets confirmed when you go to the next one, which covers that full region, and that also rescues. And then when you go further, you lost the dumpy portion, but then you have this portion and therefore let60 is there. So then you get to know it is somewhere in this portion is let60. So now you take this C10F4 and look at its left side, what kind of open reading frames are there, how many are there, and then each open reading frame you inject, then you will find the one. So in this particular case, when they did and sequenced that open reading frame that rescued, and that encoded a protein that was known already as RAS and was known only as an oncogene, where its expression caused cell proliferation, but people did not know what it normally did. And this told that RAS is actually involved in normal growth and development, and its mutation will be, we did not get into all the details because we are only using this as an example to get at the fundamental concepts, but let 60 when they identified a loss of function allele, it was lethal. Okay, so it is a recessive lethal, it is absolutely essential for embryonic and larval development. So therefore, RAS is not going to cause cancer all the time. Without it, you are not even going to live, you are not even going to exist. Okay, only when its expression is out of control, you get cancer. So that is how they discovered the growth and development role of RAS. Okay, you got an idea of how you would map a gene, right? So I will tell you a little, this is not the only way, there are multiple ways. I will give you one example. What if after while doing this, I realized that the DNA injection for whatever reason is not rescuing, okay, and a fragment of chromosome that you have taken as open reading frame or as a cosmate insert need not always express, okay, when taken out of the chromosome context. So how do you proceed further? So one approach people used to do is they make antisense RNA. Okay, so whatever is the copy of normal mRNA, now you are suspecting a particular open reading frame, and for that particular open reading frame you make antisense RNA and inject it. So at that time, so I am talking this like early 90s to almost late 90s, okay, let us say mid 90s. During that period, this was a common practice. So in plants, even they made commercially grown crops where antisense RNA expressed against a particular polysaccharide hydrolyzing enzyme allows the tomato to mature but not to ripen, because when the polysaccharide is degraded, you get monosaccharides which makes it sweet, and then it becomes softer and really becomes ripened tomato. So they use antisense to block that particular enzyme polygalacturonase expression, and it worked, it is proven and it is commercial. So when in C. elegans for one particular gene that affected the early embryonic development, like, so you remember the first division is asymmetric, one cell is larger, another cell is small. So there was one lab that was focused on finding out genes involved in making this asymmetric division. So what they did is they mutagenized and looked for mutants, like the way our mutagenesis screen. Only thing is, instead of looking for bag of worms, they were looking for dead embryos. And then once they found dead embryos, then detailed observation, like what we did by looking at the PNP fate, whether they become primary, secondary, etc. They looked at when does the embryo die, at how many cell stage. Then they find whether that dead embryo allele was actually having problem in asymmetric division. So they found one gene that when mutated led to symmetrical division, the first division. So they did this standard experiment, what we are talking about is the only standard at that time, and it did not work. So they decided they suspected an open reading frame and they decided to make antisense and inject, and it worked. So in the wild type, blocking that one particular ORF by giving antisense led to symmetrical division. So they were convinced, they mapped, but then usually people do a control is sense. So if you inject the sense RNA, you should not get that effect. So if you get that effect, then you think the effect is non-specific, maybe just injuring the gonad caused that. It could be anything, or any RNA sequence could do that. So they did the sense injection, and sense also had the same phenotype. So they got paranoid, and that is when Andy Fire, who worked in Philip Sharp's lab and the lab that won Nobel Prize for discovering splicing, he knew very well about what happens when you synthetically make RNA. Okay, so RNA chemistry he knew very well. So he reasoned out that when you are making single strand RNA, you are probably making trace amounts of double strand RNA, and that is probably somehow it interferes, and that is how RNA was discovered. Okay, so while mapping a gene, This gives you an idea of how discoveries happen in science. So, they were simply mapping a gene, and while trying to fix an experimental artifact, they discovered a new phenomenon. So, then they decided to make double strand RNA and inject, intentionally, instead of having a trace contamination, that you make double strand RNA and inject it, then it works. This happened in 98. Okay. So that is how, while mapping, they found this. So, this gives you, there are multiple ways of doing it. You could either block it or you could rescue it in both ways. So, now we are moving away from mapping. So, we will revisit little bit of mapping to the very end when we have a complex screen. So, the next is, fine, I got a loss of function allele in a gene, and it is required for a particular function. Like for example, in this one example we are considering multi-vulva, right. One of them, not just let60, but there was one loss of function lin15 or whatever, one of them gave you multi-vulva phenotype. Now you want to find out additional genes involved in it. Okay. So, you still do not have the confidence that by crushing the worm and making it into a lysate, you will be able to purify the protein that is involved in these functions. So, you do not still put lot of weightage on that technology, and you want to still rely on genetics. And what you do is, you take the mutant and mutagenize and screen for new mutants in which the old mutant phenotype is lost. Meaning, can I make a new mutation and rescue the old mutant phenotype back to wild type? They are called suppressor screens. Okay. So, here you do not do just 10,000 gametes, like you do not simply screen 5,000 mutagenized worms. Here you go large number, because you are looking for a specific genetic interaction, a new mutation that rescues or suppresses the original phenotype, meaning these two genes must have an interaction. Okay. Mechanism we will not know at this point, but they somehow interact, and that somehow interaction in terms of the phenotypic outcome, we call that as genetic interaction. Okay. So, you want to find such genetic interactions. So, this method helps you to find additional genes, and that is why we call this as really a powerful method. So, here you take multi-vulva over multi-vulva, it is the homozygous for that loss of function. I forgot the gene number, but I think it is lin15, let us assume lin15. And the new mutation, new gene that you are looking for, let us say it is wild type here, plus over plus. Okay. Now, you mutagenize and self and do the same thing and look for in F2, some of them where you have the phenotype is suppressed. You are starting with homozygous. So, you are not doing any cross with wild type. So, this MUV over MUV must remain always, which means always you should get multi-vulva, and the moment you do not get multi-vulva, then you know that your new mutation is suppressing it. And again, make sure the new mutation for this phenotype follows the Mendelian inheritance pattern, like 3:1 for the single phenotype segregation and so on, to ensure that you actually have hit a single gene that is suppressing it. And here the key is to design this. I mentioned last time itself, but in this particular kind of situation where you are doing a complex screen, you will have to screen large number, and if you are going to screen large number, the mutant phenotype should be readily visible. The difference between the rest and the mutant should be obvious, then only you can identify it. It can't be like you have to mount worms on a slide and go and sit in a microscope and look at the staining pattern of something, then you will not be able to screen millions of them. In a plate, if I have 10,001, if I look at it, the mutant should jump out. So, it has to be like that. So, that is a key. So, there were several suppressors of these two multi-vulva things, let60 was gain of function multi-vulva. So, special alleles of new genes not recovered in earlier screen because they were lethal or null phenotype, now you recover. Okay. So, like people in our lab know this, because this is the only kind of screen we have done, and we found lot of alleles where if you cause a total loss of function they would be lethal, and a specific partial loss of function would have allowed its function during the early development, and then specific situation you find the defect, particularly in the background of another mutation. So, you find those kind of different alleles having different effects. You can easily imagine an amino acid substitution in one region versus another region might have different consequences on the protein function. So, that is how you get partial loss of functions. And sometimes you get new alleles of known genes, for example, LET60. Okay. Several suppressors of Lin15 multi-vulva mutation were new alleles of LET60 with vulva-less phenotype. So, when you do not have LET60, you are actually not going to make vulva. Okay. When you lose LET60 function only during vulva development. If you lose it in embryonic development, then embryos themselves like larval lethal, and that is why the term LET, larval lethal, that is what happens. So, they ended up finding that some special alleles of LET60 made it not doing its job during vulva development. So, therefore, whether Lin15 is there or not, it did not matter, LET60 stopped the vulva from developing, and that is how it ended up suppressing Lin15. So, these are the kind of alleles that you get when you do the suppressor screen. So, these kind of variant screens, there are many different screens. So, we are not going to go through all of them because we just want to get some good idea of genetics to continue on our development. So, this is just one example of a complex screen. So, after this sort of a screen, they ended up discovering this for LET60. Complete loss means it is lethal, and antimorphic, it is dominant negative, like vulva-less, it does not allow the cells to be in the uninduced fate. Hypermorphic, if it is continuously on, then it makes multivulva. Okay. So, that is a suppressor screen. So, here what you do is your original mutant gave you a weak phenotype, not a strong phenotype. Then you want to mutagenize and look for new mutants that give a stronger phenotype, or sometimes the original mutant did not have a particular phenotype you discovered because of some other phenotype defect, but when you combine with another mutant, okay, mutation in another gene, the double mutant gives a new phenotype. So, those are called synthetic phenotypes, and a genetic screen looking for those kind of phenotypes are called a synthetic screen. So, here I am using an example. This actually did not come from a synthetic screen, this is something that I did, and therefore it fits as a good example, so I am telling this. So, but for RNA probably we would have done that kind of a screen, and I may have taken a little longer to finish my postdoc, but RNA got this story done fast for me. So, here you take an example like NOS1 and NOS2, these are closely related genes. Okay. So, in the wild type when you have both of them, it is fertile, it is totally fine, and you have mutation in NOS2, let us say NOS2 expression is extremely low. So, the activity in y axis, and NOS1 is fine, it is wild type, and the worm is again fertile. Now, you do the reverse, NOS1 is activity is reduced and NOS2 activity is high, and it is still fertile, and now you bring both of them down, it is sterile. So, this is an example of a synthetic phenotype. Okay. So, in this situation what you are going to say is these two seem to function redundantly. Okay. So, like suppressor mutation, synthetic mutation help us identify genes that function in a single pathway. So, here usually redundancy is what you are discovering in this kind of an example. Alright. So, we stop here, and in the next class we will go on to find the hierarchy or the sequence in which a given set of genes function. Okay, like for example, you found 20 genes involved in vulval development. 2 of them were multi-valva, okay, and out of which one was dominant allele. So, we are, let us say we ignore those 2. Then you have 18 of them giving valvaless. Now, which one of them act first and which one of them act as the 18th and which one of them is in a parallel pathway and so on, right. You want to find an order that is doable using genetics, okay. It is not always done using biochemical methods. Do you guys know how that was done? Like in biochemistry we know, right, A gets converted to B, gets converted to C and so on. How was that order determined? That is called epistasis analysis. That is what we are going to discuss in the next class. That is one. And second, to be fair, there were non-genetic methods like radioactive labeling of specific atom, like you label the particular carbon and then you see where that carbon goes. Like for example, in glucose, if you label the 6th carbon, now in pyruvic acid what is there at the end of glycolysis. So, this radioactive tracer as well as this epistasis analysis is how all the biochemical pathways were worked out. But of course, biochemical pathways were not worked out by doing genetics in C. elegans or Drosophila, they were done mostly on bacteria, E. coli, bacterial genetics. Okay, so, but the basic rules apply in all organisms the same way. So, we will stop here and continue on epistasis analysis in the next class.